All right, folks, everybody hear me okay?
Yes, sir.
Yes.
Okay, everybody see the whiteboard?
Yep.
All right, excellent. All right, so we got about 3:30 here, so we might as well go ahead and get things rolling here. So thinking forward to tomorrow's lab, things that are due, things that we have to look out for. So first thing first, I think you guys have a homework assignment due tomorrow, correct?
Yep.
All right, and then also you'll have the iron lab. So homework and iron lab are what I'll be taking up tomorrow, and I'll work up some sort of quiz for you guys as well. So let's see, probably the quiz this time will focus on the spectroscopy in the iron lab. So I think that's what we'll go ahead and focus on, and that's how I'll write it up. Okay, so make sure you look over those kinds of things. Fears law, absorbance, transmittance, all that good stuff. And some of that was on your homework as well. So you've had a chance to practice with a little bit of that. I think that's where we'll focus. All right, so what I want to do today is I want to introduce the lab that we're actually going to do tomorrow. It's a one and done lab like the last few that we've been doing. And it's actually an Enzyme Kinetics Lab. So what we're going to be doing is actually two things. We're going to analyze glucose. So in this case, glucose, which is a simple sugar, is going to be our analyte of choice. That's what we're going to be looking for in our unknown. That has the formula of C6H12O6. Okay, and actually glucose can be a naturally occurring simple sugar, or we can actually get it from the enzymatic cleavage of sucrose, which is regular old table sugar. So actually when you digest sucrose, regular sugar, enzymes in your body actually convert it to glucose and fructose. So glucose is a byproduct of that. It's actually where we get our fast energy from. All right, so that's one thing we're going to do tomorrow. The second thing is we're also going to use an enzymatic analysis to do a rate study. So we want to go back to a kinetic study. So what we're going to do is actually determine the order of a reaction with respect to glucose, okay? And we're going to do that using the method of integrated rate laws. So those are the two goals for tomorrow. So what I want to do is I want to give you a little bit of background on this whole business of enzyme and enzyme kinetics. So what is an enzyme and what does it do and why do we care? Okay, so when we talk about an enzyme, what an enzyme actually is a biological catalyst. And as you guys remember, this is again going back to dynamic stuff that you guys probably did about a month ago or something like that. So what does a catalyst do? Just in general, if I say I'm catalyzing a reaction, what am I doing?
It's a intermittent that's not consumed that usually increases rate by lowering activation energy.
Yeah, that's a pretty good way of summing it up. Okay, so basically we know that for any given reaction, let's do just a real simple one. Let's have, say, for example, reactant A going to product B. I can generate one of these potential energy versus reaction coordinate diagrams. So it's a reaction progress diagram. So that's the potential energy of reactants and products, intermediates, and activated complexes as a function of reaction coordinate. Now remember, reaction coordinate is actually letting us measure the progress of the reaction from start to finish. All right, so basically I could have A, which is my reactant at some potential energy. Then let's say it's an exothermic reaction. So we would have product B over here at a little lower energy. Now, what can we do here? Well, we can track the progress of the reaction. So we can say, for example, that A goes to B through a one-step mechanism. So basically we know that we have an activation energy hill that we have to climb to get from A to B. So you guys know that energy there between A and the top of the hill is the activation energy. That's the energy we have to overcome in order to convert A to B by way of that mechanism. All right. So as you can see, that's a pretty big activation energy we have to get up and over. So if we have a big activation energy, what does that tell you about the rate of the reaction? Would you expect it to be fast or slow?
Slow.
Yeah. So if you have a big hill you got to climb, just under normal thermal conditions, how does energy gets transferred? What happens through collision, right? So essentially what has to happen is the collisions have to be energetic enough to get A up and over the barrier. And that may not happen very often, so that translates to a slow reaction. But what if we introduce a catalyst? And what a catalyst does, I think it was John who mentioned it, is that essentially we can get an alternative pathway here, and we can have something that looks, for example, like this, that gets us from A to B. So that would be the catalyzed form. So we'd introduce a catalyst to make this happen. And what the catalyst is doing is it's providing us with an alternative lower energy pathway that gets us from A to B. And since the total energy required to get up and over the various humps is not as big, you'd expect the reaction to speed up. And that's exactly what happens. Catalyzed reaction is going to be faster than a non-catalyzed reaction because of this lower energy pathway that we're introducing. Also, as was mentioned, a catalyst doesn't get used up in the reaction. So it might actually be a reactant, but it gets regenerated, for example, in a subsequent step in the mechanism. Okay, so we say that the catalyst doesn't get used up. All right, so that's essentially how a catalyst works. And an enzyme is a very specific kind of biological catalyst. So in this case, what we're trying to do is actually break glucose down, just like your body would, okay, to get energy out. And to do that, glucose gets converted, as we're going to find out a little later, to two products. You get gluconic acid and you get hydrogen peroxide. Okay, so this normally would happen in an enzymatically controlled mechanism within the cell. All right, so that's what's going to happen. So we're going to have a very special enzyme that's going to catalyze that process and move it along. Now, the other thing we have to remember about enzymes, in terms of their catalytic properties, the activity of the enzyme as a catalyst is going to be optimized at a given pH and temperature. And for example, the catalysts that we have in our cellular mechanisms in our body, those are going to be optimized for physiological pH, which is just a little bit north of pH 7, and body temperature, right, which is what, 98.6 degrees Fahrenheit. And what ends up happening if we deviate too far away from either of those norms is that essentially the enzyme fails to work well. And in fact, if we get too far away from those norms, we can so-called denature the enzyme, which means we break down its structure so that it can no longer function at all. Okay, so that's why bad things happen to us when our body pH gets too far out of whack, or for example, if we have like a super high fever and our body temperature goes too far up, or we get too cold. Okay, all of those processes will shut down because the enzymes can no longer work. Okay, now, when we deal with an actual enzyme, we can look at it from a structural perspective. Okay, so we said an enzyme is a biological catalyst, but if we want to sort of look at the actual structure of the enzyme, an enzyme is actually a subset of a much bigger kind of biological macromolecule called a protein. So an enzyme is an example of a protein. Now, what's a protein? Well, a protein in its most general form is going to be a polymer, which is a very long chain molecule consisting of amino acids as its links in the chain, if you will. So we'll say it's a polymer made up of amino acids. Okay, so what's an amino acid? Well, all amino acids have this basic structure. If you think about the name here, amino suggests amine. And amine, oh, we've talked about amines when we talked about weak bases, right? Those are those bases that have nitrogen and maybe some hydrogens attached to them. And then what's the acid part? Well, the molecule must have an acidic part. And guess what? That's one of those carboxylic acids or organic acids that we talked about, again, back in the acid-base chapter. So one end of the amino acid is going to have the amine, so it looks like ammonia or NH2 attached to something. Then you're going to have a carbon, and you're going to have a hydrogen there. And then on the other end, you're going to have your organic acid, which you guys might remember looks like this. I'm leaving out the electron pairs here, so obviously there'd be a pair on the nitrogen, there'd be a pair there, a pair there, a pair there, and a pair there. Okay, if we want to stick all those in. Now you notice that second carbon in from the left is deficient a pair of electrons. So basically there's always something else hanging here, which I'm going to call X. And that's what's going to allow us to differentiate one amino acid from the others. So basically, everything that I encircle here is going to be common to all amino acids. They all have that very same structure. The only thing that's going to vary that makes one amino acid different from the next is what X is. Okay, so how many things can we hang off that carbon to make these things look different? Now, it turns out I think there are about 20 naturally occurring amino acids. I think that's right. I don't remember really from my biochem, but when you take biochem 1, so one of the first things you have to do is memorize all the amino acids, their names, and their structures. Okay, so it's actually an important thing to do. Now, what can X equal? Well, X can equal a bunch of different things. There are a bunch of different what we call organic functional groups. We could have, for example, a methyl group, which is a CH3 hanging off of there. I could have a thiol, which is an SH hanging off of there. I could have a benzene ring, which is a C6H5. And the list goes on and on. Okay, so 20 different possible functional groups or moieties we can hang off of that carbon there that differentiate one amino acid from the other. But from an acid-base perspective, they're all going to pretty much act the same way. So you can see that the amino acid is going to have a basic side. That's the amine. And it's going to have an acidic part, which is the organic acid there. Okay. Now, the whole idea, as I mentioned, is that because these amino acids have acidic basic properties, we can protonate them and deprotonate them. And in fact, what we can actually do is link them together, okay, to form these little links in the chain of this long chain polymer that we call the protein. So basically, what ends up happening is the neighboring amino acid comes in through the amine, and it ends up binding to that oxygen over there on the right. And you end up making what's called an amide bond that actually links these things together. Okay, so essentially what you end up having is a whole bunch of amino acids linked together to make a chain that could be literally thousands and thousands of amino acids long. All right, so that's what a polymer is. So when we get down to what a protein might actually look like, it turns out that proteins, of which an enzyme is an example, have various levels of structure. And this is going to make sense here in a little bit because we're going to find out that the not only the identity of a protein, but also its function is going to depend upon its structure. So the first level of structure that we have to think about is going to be what we call the primary structure, and that's what that little symbol means. And what the primary structure is going to define for us is actually the sequence of the amino acids in the chain. AA, of course, here stands for amino acid. All right, so that's the first thing we have to think about. What amino acids are next to their neighbors, because that's going to define to a certain degree the structure of the protein. The second thing we have to think about is the so-called secondary structure. Not sign up there. Now, what this defines for us is actually the orientation of the amino acid chain in 3D space. So let me give you some examples. There's really two common secondary structures that we can have. The first one is called an alpha helix, which you guys have probably heard of before. Okay, now an alpha helix looks like a coil. Okay, so if I actually looked at a region of the polymer, it would look something like this. It's going to be coiled up, kind of like DNA. DNA kind of does this. So you think about that double helix of DNA. It's kind of like a slinky type structure like this. Okay, so that's what an alpha helix is. The other option we have when we talk about these things is a so-called beta sheet. And the beta sheet does something a little different. So basically it kind of curves along like this, and then it wraps back around itself like this. And it does that again and again and again. And you can see what's going to kind of happen here. You're going to end up with sort of a two-dimensional thing that looks like sort of a wavy sheet of paper, if you will. Okay, so it's really kind of propagating out in both the X direction and also along in the Y direction here like this. So it really looks kind of like a sheet. All right, so imagine it as being kind of a crinkled piece of paper or something like that. Okay, now we're going to find out that whether we have a beta sheet or an alpha helix makes a difference in terms of the overall structure of the protein, as well, as we're going to find out as we move to item 3 here. Now, the item 3 is going to be obviously the tertiary structure. And what the tertiary structure really is the folding over of the secondary structures into the overall shape of the protein. I'll show you a picture of this in just a minute. We'll say to define overall shape of the protein. And when we talk about shape, what we're really talking about is configuration. That's usually the term that's used. Okay, so for example, if I take a protein, and you can think of it as this sort of long chain of amino acids that sort of make alternating alpha helices and beta sheets, for example, and I were to throw that into some water, which is a really polar environment, okay, what's going to happen is that big, long polymer molecule is going to rearrange itself so that it can optimize itself energetically. It can go to the lowest possible energy state. And by doing that, essentially, it's going to be formed what we call a globular protein. And that's exactly as it sounds. So basically, it just looks like this big glob, if you can think about this thing in three-dimensional space here. And it has two regions in it. Okay, as it turns out, the outside region here is primarily going to be made up of alpha helix. Now, the reason for that is because alpha helix tends to be polar. Okay, so it tends to be made up of more polar amino acids. So remember what we're doing. We're putting this thing in an aqueous environment, right? So we're going to surround it with water molecules here. And if I do that, obviously, from an energetic point of view, we're going to want to put polar things on the outside so that they can essentially interact favorably via molecular interaction with the water molecules. Make sense? So what that does is it kind of forces to the center the beta sheets, which have a tendency to be more nonpolar. Okay, so nonpolar things don't want to interact with water, so they're going to be forced into the middle of this globular protein. So you can imagine this tertiary structure is basically the folding over and reconfiguring of the protein into sort of a globular form, so that it can minimize all of these energy interactions with itself and also with the solvent that it's in. Does that make sense to everybody? So the long and short of it is we get a big blob, okay? So this is really where enzyme action comes into play. Because remember, the enzyme is a protein. So what I've just drawn for you is essentially what an enzyme in solution might look like, okay? So let me draw an enzyme more specifically for you. So the interesting thing about the enzyme is it's going to have that sort of globular form associated with it. But then what's going to happen is it's going to have sort of a cleft inside of it that looks something like this, okay? And the cleft is actually going to be the active site of the enzyme. In other words, without the cleft, okay, the enzyme would have no activity toward a substrate. So here's the deal. What I'm going to do is I'm going to have some sort of a substrate out here, and the substrate's going to look kind of like this. Now notice what can happen here. Let me put that in there. That's my substrate, which in our case could be something like glucose, right? So the whole idea here is that we want the enzyme to act on the glucose, to act as the biological catalyst. So what this means is that the substrate has to interact chemically and mechanically with the enzyme. So notice it's almost like a puzzle piece. So the enzyme or the substrate can work its way in here like a puzzle piece and nestle in nicely with that cleft. And then once that happens, some sort of conformational change occurs that causes, for example, bonds to be broken and formed within the substrate. And then the enzyme spits it all back out again, guess what? As product. Okay? But that could not happen unless I had that very specific structural aspect of the cleft there that the substrate can fit perfectly into. Okay, and that's all part of that tertiary structure of the enzyme. Okay, so everybody kind of see what has to happen there. By the way, this has a name. It's called the lock and key model, obviously. I think everybody can see why.
One moment.
Gotta tell Siri to shut up over here. All right, so we got a lock and key model. So the idea here is that the substrate looks like the key and the enzyme itself looks like the lock, right? So the key has to fit into the lock. Then when you turn the key, the lock unlocks, something happens, you take the key back out. So everybody kind of see how that works as an analogy there?
Yep.
All right, any other questions at this point in terms of kind of protein structure, enzyme structure, how the enzyme actually does what it's supposed to do before we start getting into some chemical equations here? Everybody okay with this?
Yes, sir.
All right, and again, this is very oversimplified. So when you get into a true biochemistry class, you'll be looking at this in much, much more detail than I'm giving you here. Okay, but now let's sort of look at the chemical aspect of this. Because again, what we're talking about here is a chemical reaction, right? So what happens is S, which is the substrate, is going to react with E, which is the enzyme. And you're going to get a reaction, and the reaction is going to generate the product, okay, or products, which we'll call P. And of course, you have to get the enzyme back again because the enzyme is catalyst, right? So remember, catalyst doesn't get consumed in the reaction. We're going to have it on both the reactant side and the product side because it gets generated. So in other words, we're going to regenerate the enzyme back exactly as it was before after it spits the products out. Okay, so now that's sort of the reaction. Now, if I want to look at this from a mechanistic perspective, and we're talking about a reaction mechanism here, here's what ends up happening. I've got the enzyme. It's going to interact with the substrate. So that's just basically just symbols for what I drew up there with the substrate coming into the cleft of the enzyme. Okay, so the picture is up there. Here it is simply as E and S. And what's going to happen is I'm going to get a fast equilibrium here. So I'm going to show this as an equilibrium. So obviously it'll have a forward rate constant and it'll have a reverse rate constant. So these are rate constants here. Okay, and this is what we call a fast equilibrium or sometimes a fast pre-equilibrium. So in other words, the enzyme and the substrate can get together and they can pop back out. They can get together, they can pop back out. So in other words, this reaction can go either way. So the substrate can enter into the enzyme and then pop out with no reaction, or it can enter into the enzyme, have a reaction occur, and then move forward. Okay, that's what an equilibrium is. Now, if it does choose to move forward, I get an ES, which is an enzyme substrate complex, which in a sense is now our intermediate. So if you think back about what an intermediate is, right, in a reaction mechanism, okay, we go back to that very simple diagram up there. See that little guy right there? That's the intermediate, isn't it? Okay, so that's something I have to make that's not necessarily stable as I move from reactant to product. So that little dip there is the intermediate that I get after the first step of the mechanism. But then in the subsequent step of the mechanism, the intermediate actually gets used up as I go to B. Okay, so that's what the enzyme substrate complex actually is. Okay, so once I've got that, now that can move forward to the product. Okay, so this moves in one direction. We'll call that K3. Okay, And that's going to give me the product that I want plus the enzyme back again. So now the enzyme can turn back around and do this all again. Okay, so basically what we're talking about here is a two-step mechanism. First, we get a fast pre-equilibrium that gets us to the intermediate, which is the enzyme substrate complex. And then moving forward, that intermediate can then break apart to give me the product plus the enzyme back. All right. And by the way, the second step is going to be the slow or rate determining step of the mechanism. All right, everybody good here? And again, just to get all this straight, E is the enzyme, S is the substrate, the intermediate, P is going to be the product. All right, everybody good there? So again, that's sort of going back and reviewing a little bit of kinetics from that chapter way back when. So let's talk about our case. In our very specific situation here, the enzyme that we care about is going to be called glucose oxidase. By the way, you can tell it's an enzyme because it has that ASE suffix. Anytime you see ASE at the end of a chemical name, you know it's an enzyme. Okay, so if I see glucose oxidase, I know that's going to be an enzyme that does something to glucose. All right. Likewise, my substrate here is going to be glucose. And we define glucose as C6H12O6, right? I think that's what its formula was. All right. So we have two things that we want to do here. First, our first goal is we want to be able to use this enzymatic reaction to determine the concentration of glucose in an unknown solution. That's the quant part. All right, so you're going to have two unknown solutions. Your first unknown is going to be an assigned unknown, so I can do accuracy and precision. And then we're going to try something new. The second unknown is going to be your kombucha. So I froze down the kombucha samples that you guys used for the alcohol lab and the titratable acids lab. Froze them back down. They're still over there in the organic lab. I'm going to pull them out and thaw them. I'm going to give them back to you. And that'll be the last thing we do with the kombucha. So we're going to end up doing kombucha as an alternative unknown, if you will. And I want to see whether or not there's glucose in the kombucha. There should be. And if so, I want to see if the concentration after a certain dilution is going to fall on the standard curve that we make. Okay, so the kombucha is sort of the test case that we've never done before. All right, so how are we going to go about doing this? Well, basically, I already kind of gave you the answer. We're going to use a standard curve. Okay, where have you seen a standard curve before? Oh yeah, in the iron analysis that you guys are turning in tomorrow, right? We did a standard curve there. Oh, well, so we did one with the kombucha alcohol analysis. That's the other place we saw a standard curve. Okay, so we're going to make a standard curve again. All right, so here's the problem. We're going to run into the same kind of problem we did with the iron, right? And the problem with the iron is that the iron by itself doesn't absorb visible light. And again, we're using visible spectroscopy. So we could do this lab with a spec 20. We're actually going to use a microplate reader instead, but we're going to be using visible light. All right. Now here's the issue. In our specific case, we have two products. And we're going to look at the chemistry here in just a minute. And the two products we make are gluconic acid and hydrogen peroxide, which is H2O2. Now, the problem with these is both of them in liquid solution are clear and colorless. So what does that tell me about them? Well, they're like the iron. They don't absorb visible light. Okay, that's a problem. So in the case of the iron analysis, how did I get around that using visible spectroscopy? How did I get around that problem?
We used an indicator.
Well, sort of, yeah. We used kind of like an indicator. So we used this thing called 110 phenanthroline, which you're correct, it is an indicator. You can actually use it as such because it has acid-base properties. But what did it do? What did the 110?
It made a complex that was visible.
Yeah, it made a complex that actually made the iron appear that sort of deep orange, right? So the complex that incorporated the iron stoichiometrically was that nice deep reddish orange that was obviously absorbing light in the visible part of the spectrum, okay? So we're gonna do a similar kind of thing here. We're gonna couple some reaction chemistry to one of those products, specifically the hydrogen peroxide that we make, so that we can give it some color, okay? So here's kind of what the reaction sequence looks like. And you actually have it in your handout. So we'll just go over it here. C6H12O6, that's your glucose. Okay, so it reacts with some byproduct things. You got to have some water in there, which obviously you do. You also have to have oxygen, which we do because that's typically going to be dissolved in there. And then we also have to have our enzyme, which I'm going to call GOX, which is shorthand for glucose oxidase. That's the enzyme we said is going to operate on the glucose. And what you're going to make is gluconic acid, which is C6H12O7. So that's gluconic acid. And you're also going to generate, as we said, H2O2. Now, all these things are happening in aqueous solution. I'm leaving the AQs out, but they're all there. Okay, so hydrogen peroxide is the other thing we make. So here's what's going to happen. We're going to take that hydrogen peroxide and we're going to put a colorimetric reagent in there that's going to react with it. Okay, so in a follow-up step, H2O2 is going to react with this organic thing called O-dianicidine. I'm not going to worry about the structure or what elements are involved. We're just going to call it by its name. And it's in the reduced form, so I'm going to put a little RED there. So it's not the color red. That tells me it's actually in its reduced form. And then we're going to have a second enzyme in there called peroxidase. That's actually going to facilitate the reaction between the H2O2 and the O-dianicidine. And what you're going to end up getting is O-dianicidine as the product. In the oxidized form. Now, here's the deal. O-dianicidine in the reduced form is colorless. But O-dianicidine in the oxidized form is sort of a reddish brown. You'll see the color. It's kind of a dingy reddish brown color, okay? Now, we could probably leave it at that if we wanted to, but the procedure actually goes one step forward, one step further with another reactant. And that's also there to essentially denature the enzyme and kill the reaction at the end of it. So in other words, when we define what the end of the reaction is, we want to denature the enzyme, and this last step does that for us. So we're going to take the O-dianicidine that we made, the oxidized form, so that's the OX, and I'm going to add sulfuric acid to it. So if you want to kill something, sulfuric acid's a great way to kill something. I think I'm using 6 molar H2SO4, which is just basically twofold diluted concentrated, so it's still pretty potent stuff. And now what's going to happen is I'm going to protonate the oxidized O-dianicidine. So I'm just going to call it O-dianicidine. It's still in the oxidized form, just protonated now by the acid. And it turns out to be this really kind of pretty intense, sort of almost fuchsia pink color. It's very pretty. Okay. Now, as it turns out, the pink O-dianisodine is going to absorb quite strongly at 550 nanometers. Okay, so that's dead smack in the visible part of the spectrum. So that makes it actually very convenient for us. So now if you work back through this thing, what we have is this big long mole bridge, right? So for every 1 mole of O-dianisodine pink, that comes from a mole of O-dianisodine oxidized. And then the O-dianisodine oxidized here basically comes from 1 mole of H2O2. And 1 mole of H2O2 is coming from, guess what? 1 mole of glucose. So it's a 1-to-1 to 1-to-1 mole ratio throughout here. So basically, if I can quantify the amount of O-dianisodine pink that I make, that's going to correlate directly to the amount of glucose that got used up. Okay, so everybody see that? So now, since I have that relationship, now I can make a standard curve, can't I? So basically what I'm going to do is I'm going to take a look at the absorbance of the pink O-dianisodine. So that's going to be absorbance measured at 550 nanometers, which is really due to the O-dianisodine pink, but we know that's 1-to-1 to 1-to-1 mole related to the glucose. And I'm going to actually plot that as a function of a concentration of the glucose in parts per million. What I'm really measuring is the concentration of the O-dianisodine, but I can relate it back directly to the glucose. So what's going to happen? Well, I'm going to get a linear relationship if everything goes well, because this is going to, of course, follow Beer's law. So as the concentration of the O-dianisodine increases through the concentration of the glucose, the absorbance at 550 nanometers should increase proportionally, and I'll get a straight line. So if you do everything right, you'll get a nice straight line out of this thing. Okay? Now, at that point, you're going to do the normal thing. You're going to have your unknown that's assigned to you, okay? But you're also going to have your unknown glucose. You're going to measure an absorbance, right? And then from the absorbance of your unknown, you're going to be able to use the equation for the best fit line to get back to the parts per million glucose, and then recognize, of course, that you actually did some sort of a dilution to get there, okay? So you'll have an equation, y equals mx plus b. And of course, you're going to know x, or sorry, you're going to know m, you're going to know b, because Excel is going to tell you that. And you're going to substitute in for a value of y, and you're going to solve for x, just like you did in the iron lab. It's the very same thing. All right, so everybody see how we're going to do that? Now, there's a couple other parts to this. Because we're using a microplate reader, that allows us to get a lot of absorbance measurements fast. So for example, when you're messing with the Spec 20, you can only measure 1 absorbance at a time, right? You can only put 1 cuvette in there at a time and measure 1 absorbance at a time. But the nice thing about a microplate reader, and biochemists and biologists use these things all the time, is that it's basically exactly that. It's a plate. Okay, so if I were to kind of draw what one looks like, it's sort of a plastic plate, and it has little wells in it. And I think ours actually has 96 wells to a plate. Okay, so each one of those little circles I'm drawing there corresponds to a well, which is exactly what it sounds like. It's a little indented part in there. Okay, and you can actually put solution in there with a pipette. So we're going to use an auto pipette to fill the wells. That's actually the most time consuming part, is actually going from your solutions via the auto pipette to fill the wells. But once you've done this, you can have, for example, 5 or 6 or 8 or 10 different absorbances for every one of your standards. Okay, you could also do the same for your unknown. Now, why is that important? Well, because it allows us to get reproducibility. So now, if I have, for example, for standard 1, 5 different wells, okay, and I could measure all 5 of those simultaneously using the plate reader, Now I've got 5 independent measurements of standard 1. So what does that let us do? It lets us calculate a mean and a standard deviation. And I can do that for every single one of my standards and also for my unknown. Now, what does that let me do? Now it lets me actually come to my plot that I do, and I can actually put error bars on this thing. So now that tells me something about the precision of the measurement at every single one of my average values. So here the error bar corresponds to plus or minus 1 standard deviation. All right. So that's one thing we're going to do. We're going to put error bars on here, and we have to, of course, program Excel to show us how to do that. So we're going to do that for every single one of our standards. We'll also use Excel to calculate a standard deviation for our unknown. We can also do it for the kombucha, provided the kombucha fits on the curve somewhere. Okay, so this is very nice. By using the Microplate Reader, now we can get a whole bunch of absorbance measurements all at the same time. And now for our standard curve as well as our unknowns, guess what? We have precision data, which we didn't have in the case of the Spec 20. Not on standard curve anyway. You got it for your unknown, but how did you do it? Well, you actually took what, 4 aliquots of your unknown, and you serially measured them using the Spec 20, which took a bunch of time, right? So this is a very fast way of being able to get a lot of data. So in the analytical world, we call this throughput. It's the ability to use one set of measurements obtained very quickly to get a whole lot of data. All right, so everybody kind of sort of see what we're doing there?
Yep.
All right, good. So that's going to happen. Now, there's another part of this that we're going to just briefly mention. So remember, part 1, our first goal here, was to use the reaction to determine the concentration of the glucose in the unknown solution. So that's your assigned unknown in your kombucha. So our second goal is actually to be able to do a kinetic analysis of the enzyme action on glucose. All right, so specifically what we want to know is we want to be able to come up with a rate law for this thing. And what we want to be able to do is to say that the rate is equal to a rate constant K, which we're going to determine, okay, times the concentration of the glucose. Okay, and that's going to be raised to some power X, which of course is the order of the reaction. So we want to get 2 things out of this. We want to determine the rate constant, and we also want to determine the order with respect to glucose. Okay. Now, we're going to use, to do this, the method of integrated rates. Okay. Now, how does that work? What do I need to do the method of integrated rates? What am I going to need data-wise, guys? Anybody remember?
Initial concentration, final concentration, and time.
Kind of, sort of. Time is important, right? Now, but I could also use that as you sort of mentioned it and do method of initial rates, couldn't I? That's the one where you set up the table where you kind of change the concentrations in a fashioned way and see how the rate responds to that. But remember, in the case of integrated rates, we're actually going to plot the loss of a reactant as a function of time, basically until the reaction gets to equilibrium. Okay. So for example, here's what you might expect to see. And you're actually going to make plots of this. But if I wanted to look at this in terms of the concentration of the glucose as a function of time, obviously the glucose is a reactant. And of course, as time goes on, what's going to happen to the concentration of the reactant? Is it going to increase or decrease?
Decrease.
Decreases, right? Because it's a reactant. It's going away as the as the reaction happens, right? So what you may end up getting is a bunch of data points that could look like this. They may not, but basically you're going to get something that could very much look like that, okay? Because the glucose is going away as a function of time as the reaction progresses. So how do I use a plot like that to define the order? Anybody remember? How do I get the order out of a plot like that?
Using the integrated rate laws, you basically go through them until you find one that gets you a linear slope, and then you know that's a pretty good fit, even though it may not be perfect because x might not be an integer.
Yep. So we have the various possible orders. Now there's other orders besides these, but let's just assume glucose is actually going to either be 0 order, first order, or second order. Okay. So what do we plot to test each one? Okay. Somebody remind me what a 0 order plot looks like to test.
That's just a concentration of A in a way to put a variable in.
Yeah, we'll just put that in there. A is equal to, we'll just put it in its linear form. So what does it turn out to be? It's minus K, I'm going on memory here, right? KT plus the concentration of A initial, is that what it is?
Yep.
I think that's right. You guys will probably remember better than I will. Now, if that's linear, you plot that, and that's basically what I just plotted here, right? If that's linear, that tells me I got a 0 order reaction with respect to glucose. But if it's not linear, what's my next option? Well, I'm going to test for first order. So what do I plot there? Or what's the linear form of the equation?
Natural log times equals negative KT plus natural log or initial.
Yep, that sounds right. Notice that these are all in linear form, so y equals mx plus b. So it's telling you exactly what to plot in each case. This is linear mx plus b. Okay, let's say that one is not linear. What's my next option?
It'd be 1 divided by the concentration of A equals negative KT plus 1
It's
over.
positive KT, isn't it? Am I right? I think it's positive KT.
Yeah.
Yeah, it's positive.
Yeah.
Okay. So that's what you plot. So that's your Y, that's your M, that's your X, and your B. Okay. So basically, you're going to test for linearity on all of these. And the one that's most linear is going to tell you what the order of your reaction is. Okay. So you're going to plot it 3 ways. You're going to plot your data with a 0 order plot, a first order plot, and a second order plot. And you're going to pick the one that gives you the best R squared when plotted as a straight line. And you'll be able to look at it. 2 of them should be curved and 1 will be linear. That'll tell you what your order is with respect to the glucose. Okay, does that make sense? Now, just one other thing we have to think about here, just because I didn't mention it. Notice that my rate law there is rate is equal to K times glucose raised to some power X. Now, keep in mind, there's other stuff in there as well. Because remember, I had oxygen in there, and I had water in there, and I got an enzyme in there. So how can I distill all of this down to the rate just depending on glucose and not those other things? Is that something you maybe went over in lecture? How do I focus down on the glucose to the exclusion of all those other things that could affect the rate? Because they're all reactants, right?
Yeah, I'm kind of lost. I don't know.
There's a trick. It's called flooding, which is actually making sure that the concentrations of all the other stuff are in large excess compared to the glucose. And the assumption then is that those concentrations, because they're in large excess, don't change appreciably relative to the glucose concentration. So basically, they remain constant and they all get basically lumped into the rate constant, and what you end up with is something called a pseudo-rate constant. So in that sense, what I can do is just focus down on that one reactant that I want to look at, which in this case is the glucose. Okay, so that's how I do it. And then again, that's just kind of a experimental detail that really isn't that important here, but that's how I can distill this rate law down to that one thing that I really want to focus on, which is the glucose concentration. Okay, so just to wrap up here, because I know it's about time for us to go, just to wrap up here, guys, what I want to talk about on Wednesday, because by that time we'll have done the experiment, right? So what I want to talk about on Wednesday is really how we're going to take all that data that we collected on Tuesday and do something useful with it. So there's a lot of sort of mathematical manipulations that we have to go through, and a lot of stuff we have to do with Excel to make this happen. Okay, so this is really a very heavily Excel-based lab when it comes down to the data analysis. It takes you a lab period to collect it all. And then you've got all this data you got to do something with, and a lot of it is very repetitive calculation based. So what I have found works well is to kind of go over together the various steps in Excel that we need to use. So I'm thinking what we'll do on Wednesday is kind of go over, without actually loading up Excel, maybe we can and actually look at it, but kind of just go over the steps in Excel that we would have to go through to make all of this happen. And then what I'm thinking is the following Monday, which is when this is all due, it'll all be due the Tuesday, is that it might actually be worth it for us to all, I don't know how you guys feel about it, but to actually get together face to face and go into 310 and bring your laptops with you or whatever device you use to do Excel on and kind of work on it together. And that way, when problems crop up or if somebody gets lost, you guys can help each other. Okay. Sometimes in the past, what I've done is I've kind of put a leader up at the actual computer up front and actually project on the screen what's going on. And then that person can throw their data in there and kind of help lead everybody through what has to happen. And of course, with everybody else pitching in and helping out and saying, okay, here's what I think we got to do. No, maybe we should do this other thing. So how does that sound to you guys? Does that sound like something you'd like to do?
Yeah.
I mean, we're down to like 10, 11 people in the lab. So I don't think there's any issue from a COVID perspective putting us in FS3 in 310. And that way I figure we can just kind of work on it together. And I've done that in past years. It seems to work well. And you'll have all the tools actually after Wednesday and tomorrow to actually start working on this. So what I'd suggest you do is do exactly that. Start working on it. And then when you get to the point of Monday, you can sort of check your answers against everybody else's and make sure your Excel spreadsheet is done correctly. And that way, when it gets down to actually presenting the data for me on Tuesday, all that'll be done. Okay, so you won't have people coming in saying, well, I didn't know what to do with Excel, because it'll all be there. Okay, so as long as that's okay with you guys, I think that's what I want to do. So we'll talk about it on Wednesday. And then what we can do is meet the following Monday in person in 310 at 3:30 and go through it. Okay, so if that sounds good to you guys, that's what we'll do. All right, any other questions? Everybody good?
Yep.
All right, so I'll see you guys all tomorrow. Don't forget about your homework. Don't forget about your quiz, and be ready for that lab. I will see you then.
All right, have a good one.
All right, you too.